Hello everyone, welcome back to the hematology point. Today we are going to have the process of white blood cell formation, and this is leukopoiesis. Like any other process of cell formation, the process also begins with the hematopoietic stem cell, which you can also call the pluripotent stem cell or the hemocytoblast. And this cell has the ability to differentiate into all cell types. So today we are having it in line with white blood cell formation. The hematopoietic stem cell has ability to differentiate into either myeloid or the lymphoid stem cell. So for white blood cell, it will be differentiating into the two lineages, the myeloid and the lymphoid, because the lymphoid will give us the lymphocytes and the myeloid stem cells will give us the granulocytes and the monocyte. To begin with, let's look at the myeloid stem cell lineage. In my introduction video about blood cell formation, we saw that the myeloid stem cell can give us monocytes, it can give us all the granulocytes, and it can give us red blood cells. So in the next video of erythropoiesis, we saw the myeloid lineage as a stem cell that you can get red blood cells. So we saw the red blood cell formation, and today we are going to see how the monocytes and the granulocytes are formed from this lineage, because it gives us all of them. So the myeloid stem cell will be stimulated by the granulocytic colony stimulating factor in combination with different interleukins to differentiate into a myeloblast. Remember, it's a stem cell that is going to give us blasts that will form particular cells. So as a myeloid stem cell, it will give us blasts of the myelocytic lineage, just to say, let me call it granulocytic, then the monocyte blasts. So the monoblast and the myeloblasts will be the next cells formed after the myeloid stem cell. However, to first take the granulocytic lineage, the stem cell will develop into a myeloblast in presence of granulocytic colony stimulating factors and other leukins. This myeloblast will then further differentiate to form promyelocytes, just as you hear the word pro-myelocyte. Pro to mean before, and myelocyte is a myelocyte. So this just gives us the next stage, which is a myelocyte. The promyelocyte then differentiates to form a myelocyte. Then a myelocyte will develop to form the metamyelocyte. And at the metamyelocyte, here is a stage where we can now differentiate these cells. Because we are forming granulocytes, we must form metamyelocytes of the three: the basophilic metamyelocyte, the eosinophilic metamyelocyte, and the neutrophilic metamyelocyte. At this stage, cells are having clear staining properties of the particular cells listed, and the basophilic will be showing as a dark blue cytoplasm, the eosinophilic will show a pink cytoplasm, and the neutrophilic will be showing the blue and pink staining of the cytoplasm. These metamyelocytes will develop now into band cells, which will eventually develop into mature granulocytes. We shall have a basophil, an eosinophil, and a neutrophil. Let's now quickly see how the myeloid stem cell will give us a monocyte. In presence of the granulocytic colony stimulating factor and other leukins, the myeloid stem cell will be instructed to differentiate into a monoblast. And this monoblast is the immature form of the monocyte. So the monoblast will just eventually develop into a monocyte, which is an agranulocyte cell. It will lack granules. Now on the lymphoid stem cell, the lymphoid stem cell is going to give us lymphocytes, both T and B lymphocytes. But since lymphocytes are granulocytes, we shall definitely need the factor, the colony stimulating factor of granulocytes, as we saw in the formation of a monocyte. So the lymphoid stem cell will develop into a lymphoblast in presence of agranulocytic colony-stimulating factors and other interleukins, the lymphoblast will develop into T and B lymphocytes. However, the B lymphocyte may go to the bloodstream directly, unlike the T lymphocyte, which will be first taken to the thymus gland to develop, and then to the